Thank you for having me and have the opportunity to show our latest effort on the derivation of oligodendrocytes from iPSCs. I'm Valerio Piscopo. I work for the Early Drug Discovery Unit at the Neuro, and at EDDU, we work mainly on induced pluripotent stem cells that we utilize to model both in 2D and in 3D a number of different type of cells. I personally supervise works that have to do with glia, namely oligodendrocytes, microglia, and astrocytes. And today I really want to focus our attention on oligodendrocytes. So, as many of you might know, oligodendrocytes are the myelin makers of the central nervous system. They are the cells that produce myelin sheaths that insulate axons of the neuronal cells. They develop from their own precursor cells called OPCs. They are essential for proper nerve conduction, support axonal integrity. It is also worth noting that OPC themselves, their precursors themselves, are thought to have important biological functions, and disruption of the functionality of oligodendrocytes and perhaps, as many evidences are showing, OPCs are involved in several type of nervous system disorders, of which I mentioned the majors like multiple sclerosis and leukodystrophies, as shown by Alex before. So I just want you to focus on the complex network of interactions that lead to the development of oligodendrocytes, because this might give you a hint on the challenges that we might have when we derive such cell models in vitro. In fact, once the neural stem cells are committed to the glial phenotype, those Glia restricted progenitors are really the common progenitors that might lead to the insurgence of astrocytes or oligodendrocytes through specific precursors that have a defined set of markers. As you can notice, some of these markers sometimes can overlap, and is not excluded that even when they are committed to the oligodendrocyte phenotype, some of the precursors might revert into the astrocyte phenotype. And this is very important to keep in mind because it will be the focus of our effort to define why do we have heterogeneity in the dish. So I will mainly show you two parts that are interconnected to each other. In part one, I will show you our latest publication, our latest effort, in which we utilized a reporter line expressing a fluorophore under the important oligodendrocyte transcription factor SOX10, and how we exploited this model to improve an existing method to produce OPCs and oligodendrocytes from iPSCs. And in part two, I will focus more on the heterogeneity of the cultures that we obtain aside from oligodendrocytes. And then we can discuss on whether this is really a challenge or if we can learn something from this and take it as an opportunity to study cellular aspects that are previously um, unobserved. And then I will give you some summary, some conclusions. So I first of all want to acknowledge all the people, including Alex, Gabriele, Julien, etc., that contributed to this recent publication, in which, as I told you, using this SOX10M orange construct, we did some study on cell heterogeneity when trying to derive oligodendrocytes. We improved the existing protocol to obtain myelinating oligodendrocytes, and we did some important confirmation of previous observation relating the expression of the transcription factor SOX10 to the maturity status of these cells. So briefly, let's focus on the construct first. Just to say that this is not like a classic reporter construct that has the reporter under the control of the promoter of SOX10, but we attach the M-orange reporter to the SOX10 gene with a self-cleaving peptide called P2A. So what happens is that when SOX10 is actually translated and produced, P2A gets cleaved and M-orange gets released in the cytoplasm of the cells, while SOX10 goes in the nucleus and exerts its actions as transcription factor. We also demonstrated that this doesn't affect at all the expression of the endogenous SOX10. And once we differentiate these cells in OPCs and later oligodendrocytes, we can see how they beautifully express the reporter. And this allows us also to be able to isolate these cells for purification and studies, as I will show you in a sec. This is done via fax. So in order to differentiate oligodendrocytes, we started using a well-known protocol published by Duvaras and Fossati in 2015 that consists in mainly 4 phases, from neural induction to patterning. So you want cells to have an identity corresponding to the PMN domain at the beginning of the spinal cord. That is one of the major sites of production of oligodendrocytes in the human. Then through the action of some mitogens like PDGF, we specify OPCs, proliferating OPCs, and once these mitogens are withdrawn through the action of the thyroid factor T3 and other factors, we obtain mature myelinating oligodendrocytes. Once applied this protocol to our M-orange reporter cell lines, we first of all noticed that when we mature these cells with maturation medium that we call DMM, Duvaras maturation medium, there is a mild increase in the percentage of SOX10 positive cells. But most of all, what we notice is that essentially we obtain mainly late OPCs, with a few of them expressing the myelination marker MBP. Most of them are PDGFR alpha positive OPCs, and especially the fact that MBP always lies below the 10%, which is not uncommon because it's not really easy to obtain myelinating cells in an isolated model, since there are no neurons there to myelinate. But the striking thing is the high percentage of GFAP positive astrocytes in this culture. So the first question that we asked is initially whether we can exploit the co-presence of oligodendrocytes and astrocytes to try to derive separately both from the same progenitor. And that led to the first serendipitous discovery. So with the help of beans, we applied a known commercially available astrocyte medium to our progenitor OPCs in order to derive astrocytes, right? Well, the first thing that we notice is that when we apply this medium to our progenitors, we have a boost in the expression of our reporter, meaning that we instead of having less oligodendrocytes, with that medium, we have more oligodendrocytes. Starting from this observation, we started using different combination of this medium. This medium comes in 2 components. One is the basal medium called astrocyte medium, and one are the supplements called the astrocyte pure supplements. So finally, we found that the best combination is a 2-step differentiation protocol, in which first, under the action of the supplements, which, by the way, the composition is unknown, is protected by patent, we have an increase in the percentage of SOX10 positive cells. Once we withdraw these supplements, we introduced IGF-1 and T3 that are known drivers of oligodendrocyte maturation, and this leads to a greater percentage of substance cells. Well, not so great, but what improves really is the expression of MBP gene and the downregulation of the astrocyte GFAP gene. Also, it's interesting to notice how with the astrocyte growth supplement, we have a downregulation of PDGFR alpha, which is really a marker of OPCs. So it is normal that this decreases as MBP goes up. And you can see here how really the percentage of O4 and MBP positive cells is greatly increased by both protocols. The withdrawal of astrocyte growth supplement doesn't lead necessarily to a greater increase in the percentage of O4 and MBP positive cells. However, it leads to an improvement in the morphology of these oligodendrocytes that we can measure with shell analysis in terms of multi-branching. Mature oligodendrocytes are usually very ramified, and with the final combination of AGS and then AIT, we have an improvement in morphology. As you can observe in this beautiful image, show you the high degree of ramification of these cells and even some membranes at the end of the processes of the oligodendrocytes that might be some attempt to form myelin sheaths. So that concludes part one, in which I showed you that we generated this M-Orange reported line, and we used it to optimize a protocol to generate mature oligodendrocyte from iPSC with a greater reproducibility and a greater efficiency. In the second part, I want to focus more on how we isolated and characterized these oligodendrocytes. I will show you how we found that substance expression correlates directly with the maturity of IOLs. And then in the final part, we will focus on glial heterogeneity and how is this still present, still an issue. So first of all, with the help of Julien, our cytometry expert, we defined a set of markers, as I showed you in the first scheme, to try and isolate subpopulations of progenitor in these cultures. Because the first question we asked is whether we might have some degree of cells that don't complete the differentiation, but still lie around and have the potential to become to remain progenitors and perhaps become astrocytes instead of oligodendrocytes. So we define three type of progenitors. The first is the GRP, the glial restricted progenitor, that is a tripotent cell, usually CD44 negative, A2B5 positive, PDGFR alpha negative. The second is the astrocyte restricted progenitor, ARP, that is only capable of giving rise to astrocytes. And the third is the early oligodendrocyte progenitor, called O2A or early OPC. This is still committed to the oligodendrocyte fate, but under certain conditions is capable of giving rise to type 2 astrocytes. We also noticed that the cells that expressed the higher intensity of the substance reporter were the early OPCs. And so then we did the contrary. We started isolating the cells for the expression of substance reporter. In figure A here, we can see how we divided it in three populations. One population is the M-Orange negative. One population has a medium intensity of the substance reporter, and the other population is expressing high levels of substance. the interesting fact is that when we plate this isolated population, we live isolate through FACS and replate on electrospun nanofibers, nanofiber plate, only the SOX10 high cells are capable of ensheathing these fibers, as demonstrated by this beautiful MVP staining, in which we see that only the SOX10 high cells are able to myelinate these fibers. When we isolate these cells and we analyze their gene expression, we confirm this by seeing that only the SOX10 high express a higher level of mature oligodendrocyte genes such as CMP, MBP, and PLP1. It's interesting to notice that the SOX10 negative express the highest percentage of PDGFR alpha, meaning that there might be some OPCs that do not yet express high levels of SOX10, and so those remain behind. But most importantly, the SOX10 high cells are the ones that have the least amount of GFAP, which is an astrocyte marker transcript. So all in all, this is very interesting and confirms the fact that we have a very heterogeneous culture when we don't isolate these oligodendrocytes from the rest of the culture, and there are still some progenitors lying around. Now, we asked whether we can study this population in order to understand whether we can perhaps benchmark the amount of each subpopulation and see if those maybe might change under certain cues. And to do so, we performed a single-cell RNA-seq on both our cells in OPC medium and cells in differentiation medium. And this is a work done mainly by Taylor, Gabriella, and Alex on two different type of cells, both our 3450 SOX10 cells and other type of patient-derived control cell lines. The first thing that we can notice is that we confirm that all the aforementioned populations are present, including different type of astrocytic cells. There is a differentiated oligodendrocyte population that only appears when we push these cells to mature. And this will be the object of a work that has been submitted. We also quantified the proportion of these populations and their gene expression. So one thing that is striking is the fact that the population of astrocytes seem to increase over time and then abruptly decrease once they differentiate, but the amount of astrocyte progenitor increases over time for some reason. It's also interesting to notice that there is always a population of resting OPCs that seem to be more or less constant over time. What really decreases once we induce differentiation is this population of proliferating OPCs that obviously become then mature oligodendrocytes. Then we finally tried to identify what are the developmental trajectories once we start from glial restricted progenitors that lead to the different subpopulation. And we identified three trajectories. The first one, obviously passing through astrocyte restricted progenitors, leads to mature astrocytes that are present in three populations, but we won't discuss this today. What is interesting for us is this divergence between one trajectory that leads to resting OPCs and one trajectory that leads to mature oligodendrocytes. So this is interesting because it appears like we have a reservoir of resting OPC that seem to have some role in the homeostasis of the oligodendrocyte population in general. So the interesting fact is that we identified what are the genes that drive the divergence versus the lineage 2, the resting OPCs, and lineage 3, the mature oligodendrocytes. And it's interesting to notice that none of these genes are previously observed in oligodendrocyte or described in oligodendrocyte, although they are present. For mature oligodendrocyte, mostly those are genes that are related with adhesion, migration, but also with ubiquitination, like in the case of RNF144 alpha. Whereas for resting oligodendrocytes, obviously these are cell cycle repressors previously observed in cancer models. So all in all, to conclude, we characterized this subpopulation in our iPSC-derived oligodendrocytes. We identified what are the developmental trajectories leading to the heterogeneity in our culture, and we observed a divergence between this population of resting OPCs and more proliferative subpopulations that instead are driven to mature once given the right cues. We defined what are the key genes driving this divergence, and this insight will be used by us not only to improve the culture purity in future iterations of the protocol, but we will also be able to study glial homeostasis and the interplay between astrocytes and oligodendrocytes, both in health and in disease. And having said that, I want to acknowledge all the people that were involved in this work. And without further ado, I'll take questions. Thank you.